Hello everyone, welcome to Brain Blitz Audios. In today's episode of NEET Preparation, we'll be dealing with previous year questions of biology in medical entrance examinations in India. Today we're going to be looking at the 6th chapter in the grade 12 syllabus for CBSE. The name of this chapter is Molecular Basis of Inheritance. Let's look at our first question. The process through which the amount of DNA, RNA, and protein can be known at a time is called autoradiography, tissue culture, cellular fractioning, phase contrast, microscopy. So let's look at what each process does. So let's start with our last option, option D, phase contrast microscopy. What does this do? It views living cells, microorganisms, etc. So that means option D is incorrect. The reason is that phase contrast microscopy does not have enough resolution to view DNA, RNA, and protein to its constituent layers. So therefore, option D is incorrect. What about option C, cellular fractioning? Now, cellular fractioning, as the name suggests, means to separate cellular components. So it's just to separate the various components to their respective constituents. So therefore, it does not have anything to do with the gauging of the amount of DNA, RNA, and protein. So option C is incorrect. What about option B, tissue culture? Tissue culture is the growth of artificial plants from plant tissue, and this growth happens in vitro. So that means it happens in lab conditions. So this is basically a new way of vegetative propagation. So option B is incorrect. The right answer is option A, autoradiography. So what is autoradiography? It is the use of X-ray film to detect radioactive material. So how does this help in gauging the amount of DNA, RNA, and protein? Well, DNA and RNA are both made of phosphate bonds in their backbone structures. So if you can replace the phosphorus with radioactive phosphorus, and in proteins, if you can replace the natural sulfur with radioactive sulfur, radioactive isotope of sulfur, you'll be able to gauge the amount of DNA, RNA, and protein using autoradiography. So using the X-ray film, we can take out the radioactive material which contains DNA, RNA, protein, and we can make DNA, RNA, and protein radioactive by putting phosphorus in case of the nucleic acids and sulfur in case of protein and make them radioactive. We put the radioactive isotopes of phosphorus and sulfur. And that's how we use autoradiography to gauge the amount of DNA, RNA, and protein at a time. Option A is the right option. Next question. Balbiani rings are found in A, polysomes, B, polyteen chromosomes, C, autosomes, D, nonsense chromosomes. So what are Balbiani rings? Now these Balbiani rings are temporary swellings, and these are found in a special type of chromosome that we'll be finding out, and these contain uncoiled active DNA present, and this DNA is basically transcribing RNA in the presence and at Balbiani rings. So let's look at each of our options. Option A, polysomes. Now this is a chain of ribosomes that are undergoing translation. So in translation, the ribosomes clump together around an mRNA strand, and therefore that structure is called a polysome. Polysomes are not related to chromosomes, while Balbiani rings contain DNA, so they are bound to be present in chromosomes. What about option D, nonsense chromosomes? Nonsense chromosomes mean just that. They have no sense genes, so their genes cannot be coded. No coding. And Balbiani rings contain uncoiled active DNA, which are transcribing to RNA, so there is definitely coding happening in Balbiani rings. So option D is not the right option. It's incorrect. What about option C, autosomes? Now, the term autosomes refer to the chromosomes inside the body, which are not determining the gender of the individual. However, autosomes can be both normal and they can be abnormal. They can have a normal amount of DNA, they can have a whole lot amount of DNA. So therefore, when you say that a chromosome is an autosome, you're merely saying that it does not participate in gender determination. So therefore, we cannot say whether Balbiani rings are present or not. So option C is not a viable option. It's incorrect. The right answer is option B, polyteen chromosomes. Now these are the chromosomes with large amount of DNA. And when there's too much amount of DNA, they form these temporary swellings called Balbiani rings. And these Balbiani rings give you the uncoiled active DNA, which is transcribing RNA. Here's a picture of a Balbiani ring right here. Now, let's look at this question. In a DNA helix, cytosine is paired with guanine by what kind of bond? A covalent bond, B phosphate bond, C 3 hydrogen bonds, D 2 hydrogen bonds. Let's look at each of our options. Option A, covalent bond. Now, covalent bonds in DNA are usually found in secondary and tertiary structures of DNA, and these are mostly between the sugars, hydrogen bonds between sugars, while cytosine and guanine are nitrogenous bases. Both are nitrogenous bases. So the possibility of a covalent bond between them is incorrect. Next, phosphate bond. Now, a phosphate bond is the bond which joins two adjacent nucleotides. So a nucleotide contains this nitrogenous bases, they have the sugar, and they have a phosphate with them. So one of the phosphate of one nucleotide bonds with the OH group of the other nucleotide. So therefore, they form a phosphate bond. However, the bond between the two nucleotides, which are opposite to each other in a DNA helix, is the bond that is between cytosine and guanine. So therefore, it is not a phosphate bond. What about option D, 2 hydrogen bonds? I mean, hydrogen bonds are present between opposite nucleotides and they're present between nitrogenous bases. Here's a catch, however. In option D, it says 2 hydrogen bonds, and that happens between adenine and thymine. It's A double bond T, always. So option D is incorrect. The right answer is option C, 3 hydrogen bonds. Cytosine is paired with guanine by 3 hydrogen bonds in between them. Here's the structure. Guanine having 3 bonds with cytosine. And that's why we have G triple bond C. Let's look at this question. Which RNAs pick up a specific amino acid from the amino acid pool in the cytoplasm to ribosome during protein synthesis? So which of these RNAs, they help in the formation of proteins by picking up amino acids and joining them together to form polypeptides. So these are our options. tRNA, mRNA, rRNA, and hnRNA. Let's look at each of our options. Option B is mRNA. mRNA stands for messenger RNA. And what is the function of this messenger RNA? Now, this is made by the transcription of DNA, and this provides the codons that the specific RNA uses to pick up an amino acid pool. Now, this only gives you the codons. It does not take part in picking up an amino acid and forming the polypeptide. That is done by another kind of RNA. So option B is incorrect. What about option D, hnRNA? The truth is, hnRNA is an incomplete form of mRNA. So when just after the process of transcription, you get hnRNA, which has coding sequences called exons and non-coding sequences called introns. And after sorting out these differences by removing the introns, you get the complete mRNA in eukaryotes. So option D is incorrect. What about option C, rRNA? rRNA stands for ribosomal RNA, and this kind of RNA is not involved in coding. That only leaves us with one kind of RNA, that is tRNA, which is the right option. If you remember, the structure of tRNA is a inverted loop, is a loopy shaped thing in 2D form and an inverted L in 3D form. So tRNA has a 3' end. At this 3' end, it picks up the amino acid and then forms the polypeptide as the ribosomes move along. So therefore, tRNA is the right option. Now let's look at this question. The structure of DNA is, and when we're looking at structure, this is the primary structure, the basic structure of DNA, the one that is formed by a small combination of nucleotides. So that structure of DNA is what we're looking for. So option A is linear, option B is double helix, option C is single helix, option D is triple helix. Now, when it comes to genetic material, there is no genetic material with the triple helix structure. No normal genetic DNA has a triple helix structure, so option D is incorrect. What about option C, single helix? Now, single helix is usually found in RNA. Ribonucleic acids are usually not paired, so therefore they usually have a single helix as a structure, and that's why they're more able to mutate. So they can mutate faster because of their structure. Option C is incorrect. What about option A, linear? Well, a linear structure is found in chromatin, and this chromatin is formed out of an amalgamation of DNA. So in DNA, they are packed with histone proteins and with a histone octamer, and then after that, the histone octamer forms a long chain, and a polymer of that long chain is the chromatin material that you see in the nucleus. So that chromatin material is linear. However, that is not the structure of DNA. So option A is incorrect. The right answer is option B, double helix. This is the famous model proposed by Watson and Crick. They discovered it and they proposed it. So this double helix structure is the primary structure of DNA. Now, let's look at this question. Transposon was discovered by A. Sutton, B. Strasburger, C. Fisher, D. BMC, B. McClintock. So what is a transposon? A transposon is basically a kind of gene known as a jumping gene, which can change its location inside a chromosome or inside a DNA sequence. And this is found in the maize plant. We need to find out who first found it in the maize plant. Now we have 4 options. Let's look at each of them. Option A, Sutton. Sutton is usually associated with Bovary, and they told that the chromosome was the carrier for genetic material. So carrier for genetic material, this was proposed by Sutton and Bovary. So option A is incorrect. What about option B, Strasburger? Strasburger worked in the field of nuclear division, mitosis, meiosis, those fields. So Strasburger was associated with nuclear fission. Option B is incorrect. What about option C, Fisher? Fisher discovered genetic variants, the different kinds of alleles and genetic traits that we have. So those were the work of Fisher. So option C is incorrect. The right answer is option D, B, McClintock. McClintock was the person who first found the jumping genes in maize. So option D is the right option. Let's move on to our next question. A root cell of wheat has 42 chromosomes. What would be the number of chromosomes in the Synergid cell? So in order to solve this question, we need to know the ploidy of a root cell and a Synergid cell. So a root cell is any old cell found in the root of a plant, and most cells in plants are diploid or 2N. So that means they have 2 genomes in them. So the value of 2N is equal to 42. So N is 42 divided by 2. Simple division, you'll get 21 as the answer. So we know that the value of N is 21. Now we need to know the ploidy of the Synergid cell. Now Synergid cell is formed after meiosis inside the ovum. Inside the embryo sac, inside the female reproductive system of plants, of angiosperms. So Synergid cell is formed after meiosis, and therefore the ploidy of a Synergid cell is N. It is a haploid cell. So the right amount of chromosomes in a Synergid cell would be option C, 21. Because if 2N gives you 42 chromosomes, then N has to give you half that number, that is 21 chromosomes. Let's look at this question. Okazaki fragments form a leading strand, a lagging strand, a nonsense strand, and a senseful strand. So Okazaki fragments are fragments of DNA formed during transcription. We need to find out whether the DNA formed is leading or lagging. Now let's look at option D, a senseful strand. A senseful strand refers to a DNA strand which is able to code. So it is a coding sequence. And Okazaki fragments, when processed, can also become coding sequences. So option D is incorrect because both Okazaki fragments and the other kind of DNA also form coding sequences. What about nonsense strand? Well, you can't form a nonsense strand using Okazaki fragments because these are processed to form coding sequences. So they have to be a senseful strand, not a nonsense strand. So option C is incorrect. The reason why senseful is discarded is because you can have 2 types of senseful strands, the leading and the lagging, and we need to be quite specific in terms of Okazaki fragments. Now, let's look at option A, leading strand. What is a leading strand? A leading strand is a strand of DNA which is continuous. And a continuous strand does not form fragments, so option A is incorrect. The right answer is option B, lagging strand. And this kind of strand is discrete and it is a strand with breaks, so it forms fragments. Now, the reason why is that when we do transcription, an enzyme called RNA polymerase starts the reaction before it is taken over by DNA polymerase 3. So in the DNA with 3' to 5' direction, this works perfectly because the complement of 3' to 5' will be 5' to 3', and that's the direction in which DNA polymerase 3 is working. However, when it comes to the other half, that is the DNA strand which already has 5' to 3', then the complement RNA will be forming 3' to 5'. And therefore, it does not go as smoothly as possible. So there'll be a lot of stops in between, and then it has to use RNA polymerase to start again. So basically, when you have DNA like this, one of these is continuous, and the other forms breaks like these. And these breaks are known as Okazaki fragments. And that's why they form the lagging strand, because this strand now has to be processed by removing the RNA part of it, and then you have to join it and you have to process it, etc., etc. And that's why we form hnRNA at one part of transcription. It's because of the formation of Okazaki fragments. So option B, lagging strand, is the right option. Now, this is how it's done, because when we're transcripting DNA, we're essentially breaking off the bonds in between, and then 5' to 3', it goes smoothly. 3' to 5', it goes smoothly, the DNA. However, when it comes from 5' to 3', then it doesn't go so well, and then it has to endure a lot of breaks. Moving on, Wobble hypothesis was given by F.H.C. Crick, Nirenberg, Hawley, and Trna. Now we need to find out who gave the Wobble hypothesis. So what is the Wobble hypothesis? The Wobble hypothesis says that the first two positions of a triplet codon on mRNA, and it has precise pairing with bases of tRNA anticodon. So what it essentially means is that if you have the anticodon loop here and we have the mRNA here, the first codon of mRNA and the third anticodon of tRNA, they have precise pairing. The second codon and the second anticodon of tRNA and mRNA, they also have a precise pairing. But when it comes to the third codon of mRNA and the first anticodon of tRNA, they have a weak bond. And this is the reason why we have a lot of codons coding for one amino acid. You have some amino acids that have 6 or more, we have 4 to 6 codons coding for them, and the reason is because of the Wobble hypothesis. Now, how do we know who gave this? Because this happens between mRNA and tRNA. The concept of tRNA, or in other words, soluble RNA, was given by FHC Crick. And therefore, FHC Crick also gave us the Wobble hypothesis, by which we could understand why there were many codons for one amino acid. So FHC Crick, option A, is the right option. Next question. Which one of the following pairs of terms or names mean one and the same thing? Gene pool, genome, codon, gene, cistron, triplet, DNA fingerprinting, DNA profiling. Let's look at each of our options. Now, what is a gene pool? Gene pool is the sum total of genes in any population, while genome refers to the complete haploid set of genes in one chromosome. So therefore, they do not mean the same thing. Option A is incorrect. What about option B, codon and gene? So a codon is basically a triplicate of a nucleotide. Triplicate of nucleotides. So it's basically a collection of 3 nucleotides which come together to code for one amino acid. Example, UAG, AGC, etc. While a gene is basically a linear segment of DNA. It may have one or more codons, and a gene is specific for one trait. So that's the difference between codon and gene. So option B is incorrect. What about option C, cistron and triplet? Well, triplet just means a group of 3. So like 3 nucleotides form a codon. So a triplet of nucleotides form one codon. A codon is a triplet of nucleotides. However, a cistron refers to the segment of DNA which carries information to form one polypeptide chain. So this segment of DNA gets transcribed into mRNA, and that mRNA along with tRNA forms the polypeptide chain. So for one polypeptide chain, the specific code that originated from DNA, the segment of DNA which was responsible for coding for that polypeptide chain, is referred to as a cistron. So again, these two do not mean the same thing, so option C is incorrect. The right answer is option D, DNA fingerprinting or DNA profiling. So what you do in DNA fingerprinting is you're essentially checking at the unique 0.1% of the DNA, which is completely unique to every person. So these have variable number of tandem repeats and short-term repeats as well. So therefore, by counting out the VNTRs and the STRs, you'll be able to give a profile, a DNA profile for each person. So that's why it's known as DNA profiling. And using this, we can find out whose DNA is present in this sample. Kind of like we're keeping fingerprints for identification. So that's why it's known as DNA fingerprinting. So DNA fingerprinting, DNA profiling means the same thing. It refers to the same process of identifying the tandem repeats, the repeated sequences in the unique segment of DNA, that's 0.1% of DNA, which is unique to every person. So option D, the two terms mean one and the same thing. Now, let's look at this question. What is true about tRNA? We need to find out which of these is a true statement with respect to tRNA. Option D. tRNA looks like a clover leaf in the three-dimensional structure. This statement is incorrect because the clover leaf structure, which is this one that you see, is found in 2D structure. So when you look at a tRNA in 2D, you get this structure, while when you look at a three-dimensional structure of tRNA, it looks like an inverted L. So therefore, option D is incorrect. What about option C? It had a codon at one end, which recognizes the anticodon on messenger RNA. So what it says is that tRNA has a codon which recognizes the anticodon on messenger RNA. One thing is that both the ends, both the 5' and 3' ends, are at the top and they do not come in contact with the mRNA down below. And second thing, tRNA has the anticodons, mRNA has the codons. So the codons in tRNA, the anticodons in tRNA recognize the codons present on messenger RNA, and these are present in the anticodon loop. So option C is incorrect. What about option B? It has 5 double-stranded regions. If you look at a 2D structure of tRNA, this is one structure, this is one region, this is 2, this is 3, this is 4. And here there is no double bond. It's not given. So as you can see, that's 1, 2, 3, and 4. So there are only 4 double-stranded regions in a 2D structure of tRNA. So therefore, option B is incorrect. The right answer is option A. It binds with an amino acid at its 3' end. So this is the 3' end, the longer end, and it's virtually free from any kind of double bond besides the loops. And therefore, the amino acid comes and binds here. And that amino acid has a specific code in the codon given by the mRNA. So the 3' end takes care of the amino acids, while the anticodon loop takes care of the codon matchup with mRNA. So that's why option A, it binds with an amino acid at its 3' end, is the right option. Let's move on to our final question. Which one of the following codons codes for the same information as the codon UGC? Uracil, guanine, cytosine. So now, how do you check out what is the information which UGC codes for? So we have a table of the codons present in mRNA. And as you can see over here, UGC is for CYS. So that means cysteine is the information that UGC codes for. And as you can see, what else codes for cysteine? It's UGU. So therefore, option A, UGU, is the right answer. Now, you can also think of it this way. U and G are the permanent stable codons which bind with the anticodon. So therefore, if there's a difference of C or U, you will still be getting the code for cysteine. So that makes UGU also correct. However, that also makes UGG a candidate. The reason why UGG isn't is because it is not a degenerate code. It codes for only one amino acid, and that is tryptophan. So UGG codes for tryptophan, and that's why option D is incorrect. UGA and UAG are both stop codons. So therefore, they do not code for any information. They code for the stopping of translation of mRNA. So options B and C are incorrect. So according to this table, the right answer is UGU. So UGC and UGU are the two codes. They are the two codons which code for the amino acid cysteine. So this is the table which contains the 64 codons present and each of their functions. As you can see, most of them have 4 or even 6. Most of them have 2 or 4 or 6 codes for one amino acid, and this is the reason why the Wobble hypothesis was given. It explains the reason why we have more codes for just one amino acid. It's because of that third letter. If you can see for serine, you see that U and C are stable, while this third nucleotide has a weak bond. So therefore, even if you keep U, C, A, or G, it would still code for serine. And that's why the Wobble hypothesis is still accepted. And because of that Wobble hypothesis, we have UGC and UGU coding for cysteine. So therefore, the codon which codes for the same information as that coded by UGC will be UGU. And that information is for the amino acid cysteine. So option A is the right option. That concludes this episode of NEET preparation. Now, the chapter Molecular Basis of Inheritance is very important in terms of understanding genetics, and it also is very more often asked in medical entrance examinations in India. So if you would like to review more of our questions, which we have posted in the playlist for NEET, then you can always check out our channel, Brain Blitz Audios, and you can subscribe to our channel for showing your support and for getting access to all of this useful and interesting content. You can also hit the bell icon, which is the notifications button down below, for getting the latest updates on our useful and interesting content. So until the next episode of NEET preparation, take care, stay safe, bye-bye for now.